PCR is a method for the rapid and massive amplification of target DNA region in tube. DNA amplification step consists of denaturation, annealing, and extension, and target DNA can be amplified in larger quantity through 25 to 35 cycles. However, precise quantification is difficult in endpoint PCR because it confirms the degree of amplification only after template DNA amplification is completed. So it has a disadvantage of spending too much time until confirmation. For overcoming this disadvantage, qPCR is developed. This is capable of monitoring and quantifying target DNA amplification product in real time. And there are two methods: cyber green dye and fluorescence probe. While the non-specific fluorescent dye binding between two strands of double-strand DNA, cyber green dye will bind to newly synthesized double-strand DNA during qPCR. Therefore, fluorescence intensity is increased in each cycle if DNA product is increased. And we can confirm the amount of amplified DNA product in real time. However, cyber green dye binds to every double-strand DNA, including primer-dimer, so it has a disadvantage of inhibiting the precise monitoring of target DNA product. Fluorescence probe is a high-specificity method which precisely detects target DNA containing sequence complementary to probe. Probe is an oligonucleotide with fluorescent reporter and quencher attached to its respective ends, and it will emit fluorescence as being broken down by 5' to 3' exonuclease activity of DNA polymerase during DNA amplification. Using multiple probes with fluorescent dyes of different colors, we can monitor multiple targets in one tube. PCR has continuously evolved from endpoint PCR to qPCR. However, PCR is still not entirely free from false positive results caused by carryover contamination. Via various routes including air, next PCR mixture can be contaminated by previous PCR product. PCR carryover contamination can be prevented by using UDG and DUTP. UDG hydrolyzes the N-glycosylic bond between deoxyribose sugar and uracil DNA. In the first PCR, DNTP containing DUTP instead of DTTP is used. When UDG is reacted during 10 minutes before the second PCR, uracil DNA is degraded by heat. After that, new template containing DUTP is amplified during the second PCR. Inactivated during the initial PCR denaturation step, UDG does not destroy newly synthesized uracil DNA. Newly synthesized PCR product containing uracil is destroyed by UDG again before the start of next PCR. Therefore, contamination is prevented. Now, let's begin with Enzynomics and solve every problem in PCR. Enzynomics.